Hi everyone. So this video is meant to showcase the image analysis functionalities of this collection of interactive Jupyter Notebooks, but also to save me some time in terms of training the people that are using it. We at CEMIC, the Cellular and Molecular Imaging Core Facility at NTNU, have seen that this notebook collection covers many common image analysis needs and wanted to share it with the rest of you. If you're interested in knowing more about the equipment and the services we host at CEMIC, please visit our webpage. So this tool allows you to define cell populations based on the presence or absence of multiple fluorescent markers in either multi-channel 3D stacks acquired in confocal systems or multi-channel 2D images acquired in wide field mode. In other words, you can define how many marker positive cells are in your sample. It was designed to be used with brain and organoid tissue slices in mind, but it can be applied to cell cultures or any other type of samples where you have a nuclear signal. In addition, and as part of this analysis process, it also allows you to extract average marker intensity per cell and or within each of the cellular compartments, like the nucleus or the cytoplasm. So what type of images can you input? You can use multi-channel 3D stacks where you have a nuclear signal. This is particularly useful for multi-layer cell structures where you have cells on top of each other. You can process the 3D stack as a maximum intensity projection if you have a single cell layer and do not need the 3D information. This helps in terms of computation. The analysis is run faster. And of course, you can also input a multi-channel to the image where you lack the set dimension. So what happens in terms of file formats? At our facility, we work with Nikon and Zeiss systems, so you can directly read ND2 and CZ files. You can also turn your microscope files into OME TIFF or regular TIFF format with the shape channel set XYR, and the pipeline will pick them up just fine. Just make sure you don't mix different image file types in the same folder, or the analysis might get confused. So this tool starts by separating the image into its channel components. As I mentioned before, we need a nuclear signal, in this case defined by DAPI, and we have a couple of markers, Ki67, a nuclear marker that defines proliferating cells, and NEUN, a marker of mature neurons that is expressed across the entire cell, nucleus and cytoplasm. The first step of the pipeline involves the use of Stardist to perform nuclear segmentation in either 2D or 3D. You have access to Stardist standard models to do so and some custom trained models that we train for our users. In addition, you can train your own model, but first you will need to annotate some of your data to provide ground truth to the system. All of the analysis you can perform with this tool can be done in either the whole image or in multiple user-defined regions that you can track later on. After nuclear segmentation, we use the nuclear compartment as a starting point, and applying GPU-accelerated morphological operations from Pycle Esperanto, we can simulate the cellular volume and the cytoplasm surrounding the nucleus in either 2D or 3D. This is a simple dilation operation that covers most of our use cases, defining positive cells, extracting nuclear versus cytoplasmic marker intensity. If you need a more accurate cytoplasmic area definition, I am working on doing it via CellPose, but this is a work in progress at the moment. In addition, one can easily modify these morphological operations to simulate, for example, the extracellular space. Once we have defined the cellular compartment in which our markers are present, for example, nucleus for Ki67 or whole cell for NEUN, one can start defining cell populations that are positive for said marker in 3 ways that I will cover later on. So from the total nuclei, you can extract and quantify only the Ki67 proliferating cells or the NEUN positive cells. You can also quantify and generate subpopulations if you have different intensity levels across your marker, for example, NEUN high or NEUN low. But most importantly, you can define cell populations based on the presence or absence of single or multiple markers by modifying a simple dictionary structure. Okay, So which are the three methods that one can use to define which cells are positive for a particular marker? Let's use NEUN as an example, the most straightforward one. So this first method is called average intensity measurement with min-max thresholding. And what it does is summing up all the pixel intensity values contained within the cellular compartment of choice. For example, nuclei, cytoplasm, or in this case, the cell. And then it divides it by the total number of pixels that are contained in that compartment, being a 2D shape or a 3D volume. And what it effectively does is obtaining the average intensity for each of the cells in this case. Then you get a histogram of the distribution of those average intensity values, and based on that you can set some minimum and maximum threshold within which your cells should be considered positive. So in this case, we see that most cells in the full image are negative for NEUN signal, corresponding to this background peak. And then we see a couple of flattened Gauss distributions that correspond to the NEUN low and the NEUN high subpopulations. Based on these values, we can establish two subpopulations, although we will see that these particular values are not ideal in the next slides. Okay, so let's take a look at the two subpopulations that we defined. Ideally, we would want NEUN low cells to be these cells over here, and then we would like to have NEUN high probably as these cells over there. If we take a look at the values that we used to define NEUN high cells, it was 80 as average intensity and above. So that's why we see these cells being classified as NEUN high. They have like 102, 81, 85, 90 as values of average intensity. But then we are missing these cells over here, which are classified as NEUN low. What this means is that we should decrease a little bit the thresholds that we were using and probably set NEUN low to be in between 20 to 70 and then starting the NEUN high instead of from 80 from 70. So this involves a little bit of trial and error. But then you would see why the rest of the cells are classified as NEUN low. For example, this one has a value of 35, so then it's classified as low because it's in between 20 and 80. And if we take a look at cells that are below 20 in average intensity, like these ones, these are considered negative. So they don't appear neither in the low situation or in the high situation. So this is how this system works. So method 2 is particularly useful when you have bright and specific spots that do not correspond to your signal of interest, and you will see later on why. In this case, we want to define cal-binding positive cells, a marker that sits in the cytoplasm of the cells. The first step involves defining a manual pixel intensity threshold for what you consider cal-binding signal, in this case pixel values ranging from 65 to the max range of 255, and what you consider background. Any pixel in this case below 65 will not be considered as cal-binding signal. This is something that you can easily do in NAPARI. Be aware that we are looking at single pixel values as opposed to the previous method where we were calculating average pixel values within a cellular compartment. Then we overlay the cytoplasm labels on top of what we consider cal-binding signal and calculate the regions where both cal-binding signal and cytoplasm labels colocalize in the same pixels. If we had some bright and specific spots in our signal of interest, we could get rid of those in the next step. By performing an erosion morphological operation, we remove those unspecific small regions and just maintain the ones where the colocalization area was bigger. Now that we got rid of all those unspecific small spots, we can overlay all cytoplasm labels on top of the surviving regions and just keep the cytoplasm labels that sit on top of these surviving regions, effectively selecting the colocalization binding positive cells. One can play with the amount of erosion that is applied and also with the pixel intensity threshold to define what is a positive cell. Finally, the most interactive method is the use of an APOC object classifier. This classifier uses Python Esperanto for feature extraction, so this is GPU accelerated, and then it uses scikit-learn to perform a random forest task for classification. The way to use it is very simple. You just pan across your set stack, you create a new labels layer in Apari, and then you start annotating your populations of interest. First, you start drawing on top of what you consider negative cells. You can also select single cells. Then you move into the next population. In this case, we will be labeling low intensity NEUN cells. You see that just drawing on top of a few cells is enough. And finally, we are selecting the high NEUN intensity population by drawing on top of it. At the end of this annotation process, you can check your annotations, and based on this, you would perform a classification task using a random forest. After the classifier is applied, you would be able to detect multiple cell populations of interest, and which cells are positive, which cells are negative, and then also multiple subpopulations, but all of them based on intensity features.